Okay, well, so before we get started, I just want to remind everybody that along with Hussein Shanasa, Amber is the recipient of the Harris Moyed Outstanding Graduate Student Award for this year in microbiology and molecular genetics. And for those of you who don't know, and probably many of you don't, Harris Moyed was the founding chair of our department back in 1969, and he lasted through 1975 until he took on a higher calling, become the senior associate dean in the School of Medicine's dean's office. And Harris passed away during my time as department chair. And so at that time, we established the Moyed Award in 1999 with financial contributions from a lot of people, including Harris's widow, Elaine, his brother, and faculty and staff from the entire School of Medicine, including the Department of Microbiology and Molecular Genetics, Biological Chemistry, Medicine, Pathology, and others. And with those contributions, it's now a sustainable gift that allows the recipient, as Amber knows, to receive a substantial financial award into their own pocket. So that is, they don't have to spend it on the lab. They actually get to go on vacation or do whatever they want to do with it. And I should just mention that in Roz's annual memo that goes out to faculty asking for nominations, she states that the nominee has to have excellence in the MMG graduate program, as demonstrated by excellence in research, in their seminar presentations, and active participations in departmental scholarly activities, including participations in discussions at seminars, serving as grad student rep, and other department-related activities. And I would say that there is no more worthy recipient of the description of those characteristics than Amber, and she is certainly a very worthy recipient of that. And in addition to getting the financial award, she will, after all of this COVID stuff knocks down, she'll receive a plaque and have her name inscribed on the Moyed Award wall plaque in the MMG conference room. So again, congratulations, Amber, on this really well-deserved award.
Great.
Marion, it's your show now.
Okay, congratulations, Amber. I'm going to do a very short introduction in the interest of time, so I'm going to share my screen. Okay, so Amber has clocked in at a thesis defense talk of about 45 minutes. So, and I usually do a protracted introduction. I'm going to try to make it shorter this time in the interest of time. It's been an absolute pleasure to have Amber in my lab. In fact, I think it's an honor. She started off as my mentee, but over the 5 years, there have been so many things that we've learned together and insights made that I consider her a colleague and a friend by now. In this short introduction, I want to sort of highlight two general attributes about Amber that I think are particularly impressive, and I think you'll agree that they sort of went into her being nominated for the Harris Moyed Award. The first one is her energy and enthusiasm for science and her generosity of giving back to the scientific and the society. And the second one is how much of a self-starter she is. You don't need to prod her. And I'm going to give a couple of what I think are really impressive examples. So first, in the list of fellowships and awards since Amber has come to UCI, it's a long list. I can't go through all of it. It doesn't even include all the awards that she received as an undergraduate at Seattle Pacific University. But starting with the Francisco Ayala Fellowship as a recruitment tool to get Amber to join the UCI graduate program, continuing on to the Harris Moyed Dissertation Award. It's just been a constant stream of awards. And in fact, I think in 2017 and 2018, I asked her, I don't want to write any more letters of recommendation. Let's just do some science. I'll point out that she also wrote and was awarded an NSF fellowship for 3 years and was also a trainee on the Cancer Biology and Therapeutics Training Grant. Nearly made her goal. She had a self challenge to support herself for the 6 years through graduate school, and she almost made it 5 years. There are two awards I want to highlight. One is the Stanley Behrens Fellow Award. That was particularly meaningful to Amber and myself. And I know Mr. Behrens is zooming in on this, and we're very thankful that you're coming to Amber's thesis defense. And I want to thank you for supporting graduate students at UCI. It makes a huge difference. And I also want to point out the Beckman Coulter Art Scholar Award. Again, a very meaningful award to Amber, and really want to thank all of the members of that program committee for recognizing her talents. In terms of giving back, again, I can't go through the whole list, but you'll see that Amber really spent a lot of her supposed free time being co-captain of a fundraising team for cancer research, mentoring and running the program for the, working with Nita Driscoll and Christina to invite high school students from throughout Orange County for a 6-week experience at the bench, numerous other committees, and including also, again, getting back into the community outside of campus, functioning as a science fair judge and scientist ask a science night. So that's the energy, enthusiasm, and giving back. What I want to end on is the self-starter part of Amber. So in addition to all of the courses and requirements for graduate school, Amber sought out additional experiences and training, and I think it's impressive just to let you know what those are. Within the first year of arriving at UCI, she applied for and was awarded to be one of the fellows for ELIS, which is Emerging Leaders in Science and Society. Only 18 graduate students across the entire United States were chosen. They came from all walks of scientific life, and they were charged with, so truly transdisciplinary group, and they were charged with identifying a problem in society, and then forming a group that would travel around the United States for a year, holding town halls, discussion groups, and other public fora to learn from society, but then also come up with solutions in a white paper, which they did. They presented in Washington, D.C. in December 2016. And you remember 2016 was the year that Flint, Michigan was declared a state of emergency for lead in the drinking water. So it was a timely topic. Amber learned a lot about leadership, public speaking, and working within a very multidisciplinary group. The second experience that she went for was to actually be trained one-on-one in human organoid cultures. With the help of Eric Stanbridge, who met up with Hans Cleavers at a meeting and brokered a visiting scientist position, I sent Amber to the Netherlands to learn one-on-one how to make organoids from donated tissue. It's not trivial. It's not simply reading a protocol on a piece of paper. It really is a hands-on kind of training technology. And you'll see in Amber's talk how that's been very useful in a collaboration with Mekong. The third I'll just briefly mention is something that I think Amber would recommend to any graduate student, which is Bree McWhorter's Activate to Captivate, being trained in the art of presentation, public speaking, and explaining complex concepts to non-scientists. I think Amber would highly recommend this to any of the graduate students. And finally, I'll end with bioinformatics. You know, Amber came here 2015, that was her first year. And there really wasn't any formal coursework on training in bioinformatics. And then we still struggle with this today. And it became clear through Amber's thesis work that she needed bioinformatics skills. So the self-starter Amber basically crowdsourced training time or one-on-one meeting times with a lot of our bioinformatics people. They're a wonderful bioinformatics community here, and they were generous. So Jenny Wu and Melanie Oakes of the GHTF, Harry Mangolem, and then members of the Lawson Kessenbrock Laboratory, Nick Kerrigan, Kui, hope I'm not forgetting someone, were really generous with their time to meet with Amber. But that was the basics, that was the start. And really, Amber took online courses. She just troubleshooted writing her own code. The self-starter Amber basically took that base knowledge and did even more with it. And I would consider her to be a bioinformaticist of some skill today. In fact, in the last year of her graduate work, when the COVID calamity came, research was shut down, labs were shut down in March of this year. And what Amber did was, in addition to finishing data analysis, basically worked with her husband, Tyler, to write a new kind of application that she calls Gecko. She won't be able to present it today. Geckos basically a way to visualize gene expression changes on a gene by gene basis using some visualization applications that are usually used for single cell sequencing. As she wrote a paper with Tyler, they developed all the code for this. It's a web-based browser app that even I can use. And the paper is just undergoing some minor revisions before sending back. So that'll be another first author paper with their husband, Tyler. So I urge you to check it out. I might paste the URL in the chat box. She and Tyler are doing some last minute tweaks, but you can still, if you have an RNA-seq, bulk RNA-seq data set, it's a copy paste of the data into the web browser, and you can explore all the different ways to visualize data. Okay, so that's what I have for the introduction for Amber. It's time to turn it over to her. In the interest of time, I'm going to ask you to hold your questions to the end or type them in the chat box, because Amber's, we want to give her full opportunity to talk about her work. If you get lost, you have a burning question, and you please unmute yourself and ask. But for those questions that can be held to the end, please do so. And I will either read them out of the chat box, or if there's plenty of time available, you can unmute yourself and ask Amber those questions. Okay, so Amber, I'm going to stop and
Okay, thank you, Marian, and thank you, Bert. Okay, can you guys all see my screen okay?
Yes, we can.
Perfect. All right. I really want to start off by thanking all of you for being here today. This is never really how I envisioned my defense would go, but we're making it work, and I'm excited to share some of my data with you guys. And I do want to start off with kind of a basic introduction to the human intestine, especially because this is a more diverse audience. And so once food is digested in your stomach, it first passes into your small intestine and then into your large intestine or colon. And the primary goal here is really the absorption of nutrients and water. And my research in particular has been focused on the colon, and that is largely because of the enormous amount of colorectal cancer cases compared to the small intestine. And what I'm showing you here are the number of new cases predicted in 2020, as well as the number of deaths. And you can see this impacts quite a few number of Americans. And if we look across the country and across many time points, you can see that more than 4% of Americans will develop colorectal cancer at some point in their life. And based on this graph, you can see that it increases greatly as you age. And this is one of the reasons it's very important that you get your colonoscopy age 50. Now they're perhaps saying age 45, because this is really the best tool we have to actually identify disease early and treat it as best we can. Now, in my work, we do try to study actual human tissue as much as possible. But one of the fantastic model systems we use is that of the mouse, which is very similar to the human. Food comes into the stomach, is digested, passes through the small intestine, and then into the colon. And if we actually take a cross-section of the colon and look at it, you can see this tube-like architecture. The outer layer has a muscle which contracts to help move material along. This inner layer is stromal, and it's full of support cells, immune cells, vasculature, and this innermost layer is the mucosal layer. And this is really where my work has focused. If we zoom in on this mucosal layer, you'll see this folded structure. And each of these individual folds makes up a crypt. And these crypts are formed of epithelial cells. And it's these epithelial cells that really facilitate absorption, create this important barrier, and are very rapidly proliferating. And also of importance is that it's these epithelial cells that cancer often arises from in the colon. If we zoom in a little bit more on this colon crypt, I'm showing you here that it's made of several different cell types. And importantly, there's really 3 key different regions of this colon crypt. Down at the base of the crypt, there's a stem cell niche, which is largely composed of stem cells. And these are very rapidly proliferating stem cells that give rise to daughter cells that then move up into the next zone. And we can identify these stem cells based on gene expression of the marker gene LGR5. Now, once these stem cells divide, their daughter cells are pushed up into this next zone. They start to lose stemness, and they're essentially in a progenitor state where they've begun to commit to either a secretory or an absorptive lineage. These cells then continue to lose stemness and then further differentiate into mature cells. And this is the list of these mature secretory cells and then the mature absorptive cells that perform important functions in the crypt. One very important thing to highlight about this crypt is the rapid kinetics with which this process happens. From the time the stem cell divides, gives rise to the daughter cell, and that cell moves up this conveyor belt to the top, takes only about 5 days. And what that means is that the entire lining of your intestine is really replaced more than once a week. And this results in the intestine being one of the most rapidly proliferating tissues in your body. And one of the ways this process happens so rapidly is this loss of stemness and commitment and differentiation is by remodeling and changing the transcriptome and the proteome. So if I remind you a bit of kind of the central dogma of molecular biology, within our DNA, we have many different genes, and those together form our genome. From each of those genes, an RNA copy can be made, and I'm showing you here kind of a simplistic processing of that RNA. The mature messenger RNA then all pooled together is what forms the transcriptome. But what's important to note is that each gene can form more than one RNA. And that's done through alternative RNA processing. The types of events I've normally focused on are alternative splicing or alternative polyadenylation. But together, these create different isoforms of RNA, and as a result, can impact downstream protein and the proteome. And broadly speaking, all of these different isoforms can have quite a large biological impact. We can then study this process by doing mRNA sequencing, where we gather up all of this RNA and sequence it, and that really gives us information about gene as well as isoform expression. We can do this on both a bulk RNA level, where we gather up a lot of different cells and pool all of their RNA, or on a single cell level, where we look one cell at a time. My work has really been focusing on the idea of intestinal stemness in really 3 different settings: normal, wounded, and cancer. In the normal setting, we've been asking questions of what actually facilitates a rapid loss of stemness and differentiation. In a wounded setting, there can be a loss of stem cells, and we ask, how is homeostasis following that loss of stemness restored? And finally, in a cancer setting, we investigated whether stemness could be a potential therapeutic target in colon cancer. I'm going to first start off in the normal setting. And one of my first tasks was really to actually develop tools to isolate all of these different cell types within the crypt. And we did this via flow sorting. We started with a mouse colon, cecum to rectum, and did a mechanical dissociation to physically pop out the crypts. Just to show you what this crypt structure that I've been talking about actually looks like, these are microscopic images of those crypts. And once we'd isolated those crypts, we could then dissociate further into single cells and perform FACS. And FACS is essentially a way we can sort cells based off of the proteins or markers that are on them and identify those one cell at a time. This data is displayed in a FACS plot as I'm showing you here, looking at 2 distinct markers, and each dot on this plot is one cell based on the expression of these different markers. And when I began to create this protocol, this was essentially the FACS plot we would get, which kind of has a blob and no distinct cell types. But as I began to optimize this further, particularly leaving out EEE, we could actually reliably identify these 6 distinct populations. And once we could do so consistently, we were able to gather enough of these cells and populations to look at transcriptomic differences between them using bulk RNA-seq, and also look at proteomics as well. However, one of the first things we also had to do was actually determine what cell type each of these populations was. And one of the ways we could do that was actually using a very well-established and characterized mouse model system. And you can see I'm showing you down here, this is a crypt image. At the base of the crypt, stem cells are labeled with a green fluorescent protein based on their expression of LGR5. So we could use this mouse model system, isolate all of these crypt epithelial cells, and then look at which ones had the highest amount of GFP expression, which would be high LGR5, and those would really be our stem cells. And when we isolate these cells and overlay them on our FACS plot of the 2 markers I showed you previously, they all end up clustered in this top corner, which is really a perfect overlap with this population. And that really indicated to us that these were the stem cells. However, we were quite intrigued at the cells that kind of trickle into these other populations. In particular, this cohort here, which was really a rather small population. And when we actually isolated these blue cells and looked at their GFP expression, you can see most of them are negative, but a few of them do have LGR5 GFP expression. And once we had done our RNA sequencing and gone back, we could actually identify that these were tough cells. And although they don't have the same high expression of LGR5 in the stem cells, they do have elevated LGR5 expression compared to other cell types. And again, using this sequencing data, we could then look at known marker genes of all of these populations and actually identify them. We had absorptive progenitors, which are starting to be committed to the absorptive lineage, and then enterocytes, mature differentiated absorptive cells. Enteroendocrine cells, a mature secretory cell, and then a mixed population, which we called sec-PDG. And this really contained some secretory progenitor cells, as well as some more mature secretory cells. Now, one of the first things we were interested in actually looking at is comparing the transcriptome and the proteome of all these different cell types. And we were quite intrigued to see that if we were to bin based on RNA expression, we have a pretty strong relationship between RNA and protein expression, as we'd probably expect. But if you look on a gene by gene basis, you can see there's a lot of variation, which really suggests this is a complex system with a lot of additional regulation, translational or post-translational regulation. Now, using this deep sequencing data, we could also start to look at new patterns of signaling and intra-crypt communication. There's been a lot of signaling pathways that have been identified to be important in the intestine, and we were really able to, uncover some additional markers that previously hadn't been as well characterized. And one example I'll show you is that of KIT signaling, where we observed KIT receptor was highly expressed in the secretory cell types, notably in tuft, whereas the ligand was highly expressed in stem cells and the absorptive lineage. This really facilitates stem cells being able to send a signal to the secretory lineage. We can look further, we can look further and actually start to see how similar just these cell types are broadly across their transcriptome and proteome. This is using a principal component analysis, and each dot here is a sample type and shows how similar, in this case, the transcriptome are to each other. We are quite captivated by the closeness of stem and abs-pro. However, even still, we were able to identify a cohort of 16 markers that really labeled the intestinal stem cell population and were differentially expressed compared to all other cell types. For several of these markers, they were also expressed at high enough levels that we detected them in our global proteomics, and we could confirm both on the protein and RNA level that these were indeed stem cell markers. However, we are still rather captivated by that closeness between the stem and abs-pro population. I'm showing you here the data displayed a bit differently, but what this actually is looking at the number of differentially expressed genes between stem and all of these cell types. In orange are the number of genes that significantly increase versus the number of genes that significantly decrease in expression. And you'll notice again, stem and abs-pro are very tightly paired. And what was interesting is when we then performed additional bioinformatic analysis to look at changes in RNA processing, we actually observed a burst in RNA processing between stem and abs-pro. And what this really suggested is that even though there's not a huge transcriptomic difference between stem and abs-pro in terms of gene expression, the transcriptome is being remodeled by changes in RNA processing. Now, to just show you one example, I'll show you that of SPEN. And SPEN is essential in Drosophila intestinal stem cell maintenance. It plays an important role in notch suppression and Wnt activation. And the RNA processing event we are looking at is the skipping of exon 9. In stem cells, about 50% of the time, this exon 9 is included, whereas in the progenitors and the more differentiated cell, it's nearly always included, which could mean an intact RNA binding domain. Now, kind of our big question here is what actually facilitates these changes in mRNA processing? This is happening on a very rapid timescale and is really quicker than changes in mRNA expression level. what we really hypothesize is it's actually protein modification of mRNA processing regulators. And in order to investigate this further, we're continuing our collaboration with the Wiley Lab in order to do targeted proteomics as well as phosphoproteomics. Now, one of the other big questions we have is, does RNA processing actually enable intestinal plasticity? And by plasticity, I mean the ability of the cell to change cell state. And so the idea is that if these mRNA processing changes are happening during this loss of stemness, could it also enable the regaining of stemness due to its ability to switch back and forth? And one of the model systems where regaining of stemness is important is actually in wounding. So in wounding of the colon crypt, one of the common events is a wounding of the cell on the top epithelial barrier. Sorry. But this type of wounding does not dramatically change homeostasis. These stem cells at the base of the crypt are able to continue to rapidly divide, give rise to daughter cells, which then move up the conveyor belt to essentially repair and replace these damaged cells. However, the type of wounding that we've been investigating is a wounding following irradiation, where the cells that are most susceptible to damage are the cells that are rapidly proliferating. And what we see following irradiation is there is some damage to cells further up the crypt, but the bulk of the damage is focused at the base of the crypt. And the question has really been, how does repair happen here? And what's been proposed by many other researchers and supported with some evidence is that these cells actually have the ability to de-differentiate and then reform the base of the crypt and regain stemness. But the question of which cell type is most likely and how does this process initiate has not been thoroughly investigated. And so we started off by looking at flow sorting data of irradiated mouse colon, and we could see immediately 1 day following irradiation, there's a rapid drop in the number of live epithelial cells. However, by day 4, we already start to see recovery. And interestingly, by day 7, we have a very strong recovery and essentially overcompensation and increased amount of epithelial cells. And this really suggests that we already have a huge induction in proliferation. When we actually look at the structure of some of these crypts and the intestine using H&E, you can see the cecum is highly damaged and the rectum is a bit less damaged of the same mice. And the cecum, you can see here on day 1, we have a loss of this nice crypt architecture, and even further on day 2. And day 1, the base of the crypt is still intact. You can see the cells are kind of mumbled and there's not a nice clear structure. And by day 4, we've really lost a lot of these cells. Interestingly, by day 4, the base of some of these crypts has started to be repaired, whereas other crypts are still greatly damaged. The base of the crypts that have been repaired do start to look more similar to the normal, and they are now really repopulated with cells. And then interestingly, on day 7, the base of all crypts seems to have been repaired, and you can see how tightly packed in these cells are. When we start to measure the length of the crypt, in wild type, we see that the cecum, and this is consistent with data from others, the cecum has a much shorter crypt length and the rectum is longer. On days 1 and 2 following irradiation, we have a truncation of the crypt length because of the damage, and this is really concentrated in the cecum. However, interestingly, on day 4, we start to see a recovery and increased length, and on day 7, we see a dramatic overcompensation and increased length, which really is suggesting an increase in proliferation. However, we wanted to start to investigate the transcriptome a bit more. And so in order to do that, we set up a single-cell RNA sequencing experiment where we had control and irradiated mice, and we harvested the EpCAM-positive colon cells just days 1 and through 4 following irradiation. I'm showing you here this data displayed in a UMAP where each dot here is a single cell, and they're arranged in space based on their transcriptome and how similar they are to other cell types. We can then bioinformatically identify these clusters, which are essentially different cell types. Now, since we looked at both wild type as well as several different days following irradiation, we can then study and investigate how these different clusters or cell type changes over this course of wounding and the initiation of recovery. And we can also look on a cell type and on a gene level. And one of the genes or types of genes we really wanted to look at right away were stem cell markers. So I'm showing you here LGR5 as well as some other stem cell markers. And you can see these are highly expressed in the wild type, still persist in day 1, which is consistent to that base of the crypt still being intact, but then are lost by day 2 and do not return by day 4, even though we do have the recovery in cells down at the base of the crypt. Now, if we have a loss of stem cells, we also wanted to see how are other cell populations recovering or how are they responding. And so we can first look at the abundance of some of these lineage neutral cells. You'll see I do have stem cells in here, and consistent with the expression of just stem genes, we see a loss of stem cells on day 2 and they don't return on day 4. We also have a decrease in unproliferative TA cells, but we get a marked increase in proliferative TA cells by day 4. Looking at secretory cells, we also observed that deep crypt secretory cells, a support stem cell, is also lost by day 2, which really suggests damaging to the entire base of the crypt. SecPro has a little bit of a decrease and then stays steady. Goblet cells are not markedly changed. And cluster 0 actually has a strong increase on day 4. And this is a population we're still investigating further, but it seems to be a type of wounded secretory cell. However, the cell types that we were really intrigued about were actually absorptive cell types. And the reason for this is in contrast to all other cell types, we observed an increase in relative abundance of enterocytes in some of these absorptive cells immediately following irradiation. And this suggests that this population, although damaged because we have a general loss of epithelial cells, that relative to others, this population is staying a bit more steady or even increasing. We then did some IHC staining using beta-catenin to look at the adhesion junctions between cells. You can see in the wild type, we have a strong epithelial barrier and many intact adhesion junctions. In day 2, you can still see this barrier seems to be relatively intact, even though we do know irradiation does induce some leakiness, but these cells look healthy and they're quite compact. Whereas on day 4, we notice a shift. We still see adhesion junctions between cells, but these cells are now a bit bloated. There, in some areas, looks like there's delamination, and this barrier does not look as healthy. And this also correlates, like I mentioned, with that drop in enterocytes on day 4. Now, we wanted to also look at what were the proliferating cells and how are they responding to irradiation, since we do see this increase in proliferation on day 4. We then isolated all of the proliferative cell types and reclustered them. We ended up with 4 kind of essentially cell types with a TA, lineage neutral, absorptive, stem, and secretory cell type. When we look at the proliferating stem cells, they're very similar to stem cells as a whole, and this is because most stem cells are proliferative. When we look at proliferative secretory cells, they're really abundant in very low levels. They decrease a bit and then stay pretty steady. We're most intrigued, though, by the increase in absorptive cells and TA lineage neutral proliferative cells. And what this really suggests to us is that these are the dominant proliferating cell types. We then stained with pCNA, since this is able to mark all of these cell populations. And you'll see in wild type colon, pCNA is really restricted to the base of the crypts. Following irradiation on day 1, the base of the crypt is highly damaged, but there is still some residual pCNA staining. By day 2, there's a bit more increase in pCNA staining, but really by day 4, when we look at a crypt that's now recovered at the base, you can see a large amount of pCNA staining, including those cells at the base. And then by day 7, we look a lot more similar to the wild type. Now, most of the data I've been showing you has really been in the colon, but I have been investigating the small intestine as well. And the structure of the small intestine is a little bit different. We have here the crypt and a very long villi. But you'll see pCNA staining is really restricted to the base of the crypt. And if you zoom in, you can see individual pCNA positive stem cells down at the base. Now, the stem cell the small intestine is a lot more sensitive to irradiation than the colon. And so we get a complete damage to all of the villi. And on day 4, there still is not as much recovery as there is in the colon. But what you will see on day 4 is an increased expression of PCNA positive cells. And in particular, I've been really captivated by this image that shows almost a string of PCNA positive cells trying to move back down into the base of the crypt and repopulating where there's this lack of cells. And what this data has really suggested to us is that it's the proliferative TA cells and absorptive cells that are emerging as this dominant group that is able to repopulate the stem cell niche. However, I haven't mentioned much about secretory cells, and in response to wounding, we do still see a secretory response, and that is really in the expression of secreted peptides. Mucins are very important in wild-type intestine and act as an important barrier in keeping the amount of bacteria in a normal homeostatic abundance. And we see that on day 2 and day 3 following irradiation, there is really an increase or an induction in some of these mucins. If you look at MUC2, for example, I'm showing you in orange the populations that are generally producing and secreting a lot of mucin. And you can see in wild type, they have a decent amount. And following day 2 irradiation, this is increased, but we also notice an increase broadly across many cell types, including non-secretory cells. And then that is lost by day 4. Another class of secreted peptide that we looked at was antimicrobial peptides. And many of these are at a low expression wild type, entirely lost by day 1 and 2, and then really strongly expressed on day 3 and 4. In these examples I'm showing you here, CMA1 has essentially 0 expression on day 2, but largely induced on day 4, both in those secretory populations, but also broadly across many cell types. And because of this interesting expression in essentially non-secretory cells, we wanted to actually stain and look at the protein level of these. And interestingly, when we do that, if we look at CMA, for example, we notice no expression in wild-type mouse, and we did see an induction in expression on day 4, but really restricted to the base of the crypt. On day 7, it's no longer at the base of the crypt and has started to move up the crypt a bit. RET NLB, on the other hand, was highly expressed in wild type. Was lost on days 1 and 2, and then on day 4, we actually can see an induction, and it's highly secreted into the lumen of these crypts. But interestingly, when we actually looked at the regional location of these, CMA1 is really restricted to the rectum, whereas RET NLB is actually restricted to a portion of the cecum. So even though these are broadly expressed in many cell types, we really only observe them limited to a certain area and a certain time frame. And so this really suggests there's a lot of translational or additional regulation of these genes. Overall, what this really indicated is that there's 3 immediate steps in wound repair. The first is maintaining that epithelial barrier. The second is secreting of protective peptides. And the third is increased proliferation to start to facilitate the wound repair. The big question is really, how is the stemness restored? And although we don't have definitive answers, our data really suggests that TA and absorptive proliferative cells are the dominant cell type that can then repopulate the base of the crypt and would eventually give rise to stem cells, although of course other cell types are possible. We also suggest that residence in the stem cell niche is crucial for restoring stemness, and it takes a little bit of time because even by day 4, although we have cells at the base of the crypt, we don't see a return in stemness. This is unpublished work and still ongoing projects are being done, and one of the things we're investigating is actually mathematical modeling of the crypt wounding and repair, in particular the overcompensation. We're also doing additional single-cell analysis to look at the linear trajectory and looking at time and pseudotime to further investigate this idea of how is the stemness restored. Now, one of the reasons we've also been interested in wounding is the connection to cancer, and the idea that cancer is really this wound that never heals. And one of the examples of this is actually chronic intestinal wounding, such as IBD or inflammatory bowel disorder, and how many of these patients can actually have increased risks of cancer. And what this suggests is we can look at the wounding process in a normal crypt and how the healing occurs, and then study in cancer how the healing doesn't occur. And we can also investigate, just in a normal crypt, how homeostasis is maintained and how that's disrupted in cancer. And one of the microenvironment impacts that we've been looking at is actually that of metabolism. And what we've really seen, along with many others, is that cancer cells have an altered metabolism. Now, when I refer to metabolism, I really mean the energy and the food that cells are eating. And the two main sources of energy for cells are really glucose and glutamine. These are both brought into the cells through the citric acid cycle, can produce energy, and are building blocks to create all of this other cell material. But what's been observed is that in cancer cells, there's what's called the Warburg effect, whereas independent of the level of oxygen, glucose is actually shunted away from producing the maximum amount of energy and instead produces more building blocks. As a result, many cancer cells are glutamine addicted. And downstream of glutamine is glutamate. And so a potential therapeutic has been glutaminase inhibitors to try to prevent this process. But interestingly, downstream of glutamate is alpha-ketoglutarate. And it's alpha-ketoglutarate that actually feeds into the citric acid cycle to create energy. But in addition to that, alpha-ketoglutarate can also act as an essential cofactor for many chromatin-modifying enzymes. And so we really became interested in what actually happens to colon cancer under low glutamine or low alpha-ketoglutarate versus high glutamine or high alpha-ketoglutarate. And this work was initially started by the Kong Lab here at UCI and a former student, Tai Tran. However, they became very interested in collaborating with us because we had developed a human colon organoid biobank. And Marion briefly had mentioned this organoid model system, but I just want to highlight a little bit more of what this actually entails. You'll remember the crypt structure that I spoke about before, and this is actually human tissue as I'm dissecting it, and you can see these individual honeycombs are individual crypts. And once we isolate those crypts, we can then grow them as a 3D spheroid or organoid in a 3D matrix with all of the appropriate growth factors and nutrients. And just to show you what this actually looks like, these are these organoids growing in matrix. And we can do the similar thing from patient tumors. And you'll notice that the morphology of some of these is a bit different. And the reason for that is that these tumor organoids really recapitulate or recreate the tumor from which they're taken from. And so they're a very good model system to actually investigate differences in drug response or other aspects of the tumors. So one of the first things we did is we started with these tumor organoids and we treated them with alpha-ketoglutarate. And you'll notice our controls are a large, cystic, more stemmy organoid, whereas with higher amounts of alpha-ketoglutarate, we end up with a smaller, more dense organoid. And when we stain either the control or an alpha-ketoglutarate treated organoid with markers for differentiation, you'll see an increased expression of these differentiation markers, which really suggest a more differentiated, less stemmy organoid. And another important aspect of this is that this isn't a reversible effect. So if we treat it with alpha-ketoglutarate, wash it out for a week, the organoids still do not return to how they looked previously. When we expand and look at several different organoid lines, so each of the T's here is a different tumor organoid, we can see across the board there's a loss in proliferation and a decrease in organoid number after alpha-ketoglutarate treatment. When we start to look at the transcriptome and see what differences are occurring, we can see with alpha-ketoglutarate treatment, there's a decrease in Wnt target gene axon 2 expression and a decrease in our stem marker LGR5 expression. And inversely, there's an increase in expression in differentiation markers. Using another model system, a mouse organoid system, we can grow small intestine organoids in a normal amount of glutamine, and you can see this normal blebbing structure that's characteristic of small intestine organoids. When they're put in a low glutamine environment, they shift their morphology to a very cystic, stemmy organoid. Whereas with alpha-ketoglutarate, they're a very differentiated organoid. And alpha-ketoglutarate is able to essentially recover the morphology with low glutamine to have a more differentiated organoid. When we look at gene expression of LGR5, you can see an increase in stemness with low glutamine and the decrease in stemness with alpha-ketoglutarate. And the inverse is true when we look at keratin 20, a differentiation marker. And again, when we look more globally using RNA-seq data, we see that low glutamine results in increased Wnt target gene and stem expression, whereas a decrease in differentiation. And alpha-ketoglutarate has the exact inverse effect. When we wanted to probe the mechanism this a little bit more, we looked at global patterns of methylation. And across the board with alpha-ketoglutarate, we saw a drop in methylation. And when we pair this data with the bulk RNA-seq data, we can find a cohort of genes that are both upregulated and hypomethylated. And a few genes that are in this group that are actually of interest are DKK4 and 3, both of which are inhibitors of Wnt signaling. Now, the big test really is what happens in vivo. And we used an APC-min mouse model system, which develops spontaneous tumors. And when we treat with alpha-ketoglutarate, we have a decrease in tumor count. When alpha-ketoglutarate is put in their drinking water, we have a decrease in the severeness of the disease. So you can see here a drop in rectal bleeding. And we can also see prolonged survival of the mice treated with alpha-ketoglutarate as well. Another model system we can use is AKP mouse organoid. Under control conditions, this is a very cystic and stemmy organoid, but with alpha-ketoglutarate treatment, it has a much more differentiated characteristic. When we inject these organoids as xenografts into mice, and we treat with alpha-ketoglutarate either in water or via IP injection, in both cases, we have a reduction in tumor volume. So what this really suggests is that low glutamine can promote stemness, whereas high alpha-ketoglutarate promotes differentiation and can decrease tumor burden. This is really a note of caution for glutamase inhibitors to treat colon cancer, because you could potentially be driving a more stem-like cancer cell. So today I've really walked you through 3 settings: normal, wounded, and cancer. In the normal setting, we had developed a novel flow sorting procedure to really isolate all these different cell types. This enabled us to really define the transcriptome and the proteomic signatures of stemness. And the important finding was really that alternative mRNA processing changes dominate mRNA expression level changes during loss of stemness. In a wounded setting, we found that only just 4 days following wounding, there's actually a dramatic increase in proliferation. And even though stemness is lost by day 2 and the bases of crypts recovered by day 4, stemness is still not returned at that point. And we also suggest that proliferating TA and absorptive cells are really the ones to repopulate the stem cell niche. In the cancer setting, I had worked to develop a human organoid biobank. We showed that alpha-ketoglutarate treatment decreases stemness, and that decreased stemness can improve survival and lowers tumor burden. Overall, what we really show is that intestinal stemness is crucial in maintaining normal intestinal homeostasis, healing wounds, and can also contribute to cancer. And the overarching goal is to really learn how to properly control stemness to facilitate appropriate wound recovery and potentially prevent or treat disease. Now, if you remember back to the beginning of my talk, I showed you some stats about colorectal cancer and the number of new cases and deaths. However, this has been improving over time really because of new therapeutics and increasing rates of colonoscopies enabling early detection. But one of the cancers that has not improved very much is that of pancreatic cancer. So even though there's not near as many new cases, the number of deaths is almost on par with colorectal cancer. And the reason for that is that the 5-year survival rate across all stages is only 9%. And even if you don't know someone who's personally been impacted by this disease, I think this year all of us have really felt the impact of this disease. And one of the questions I frequently get asked is what's next for me? And the answer to that is New York. And I will be heading off to do my postdoc at Cold Spring Harbor Lab in Dr. David Tuveson's lab focused on pancreatic cancer to really hopefully improve the outcome for patients that are diagnosed with pancreatic cancer. And with that, I really do want to take a bit of time to acknowledge many of the people that have been involved in this journey. And of course, that starts with all of the collaborators that I've had. I'm listing here many of the people that I've worked on projects with. Many of them are co-authors. or part of different core facilities that have been so influential and instrumental really in getting this work done, as well as my committee members and many of the other advisors I have. And I can't give these people really enough gratitude, and the work really wouldn't have been possible without them. Another important thank you needs to go to all of the funding. Marion did mention that I've been able to receive a lot of different fellowships and awards, and this goes towards paying not just for research aspects, but my tuition and my stipend. And I'm beyond appreciative for all of the resources that I've been given. And similarly to Marion, I do want to highlight two of these awards in particular, the ARCS Foundation as well as Stanley Barron's. And the ARCS Foundation is absolutely a phenomenal group that I'm so blessed to have been a part of. These wonderful women fundraise, host wonderful events, and really support graduate students and our research. And it's been quite an honor to be a part of this wonderful group. I also, as Marion mentioned, wanted to thank Mr. Stanley Barron's, who, although he didn't necessarily have, a huge science background, he just really has been impassioned to be a part of science and graduate students. And I'm very appreciative for his interest and support of not only me, but all of the School of Medicine graduate students. Another donor that I've been really honored to interact with is Dr. Lorna Carlin, who also has that same heart to really support and appreciates graduate student research. The School of Medicine has been a fantastic group to be a part of, and I really want to thank all of my fellow School of Medicine students. I've also got the honor to be on the grad council for quite a few years, where I've been able to work with Dickie in one of my, you see him here in one of my favorite pictures of him, and also just work closely with him and Leora. And this is actually one of my favorite videos of him and Leora humoring me and searching through an ice bucket for little tiny clear PCR tubes, practicing for grad day. And I've just really been appreciative to be able to work with them over the years. As Marion also mentioned, I've been a part of the CRI group in addition to the Oncoslayers and hosting the high school program. And I'm really appreciative for Nita and Christina and Jamie for really keeping everything going and keeping our Sprague Hall building running and really enabling a vibrant cancer community here at UCI. I was also able to be a part of the T32 training grant, which has just been a wonderful opportunity and really facilitates that community of cancer researchers. Of course, I also have to thank my department, all of the MMG faculty, many of the MMG students, past and present. Has been wonderful to get to know them. They've always been very supportive, and I've appreciated their help in so many different ways. As well as administrative staff that really keep, everything going. There's a lot of behind the scenes work that needs to happen, and I've always been very appreciative that they've been there to help with that. I've also been a part of the CASB or U54 group, and Administrator Karen Martin is really the one that keeps everything held together on that end. I, of course, need to thank everyone in the Waterman Lab. Our lab has perhaps shrunk a bit in size in the past few years, but I've really appreciated just being a part of this wonderful community and group. I immediately felt at home when I rotated in the Waterman Lab, and it really felt like the place I was supposed to be, and it's really always felt like that. So I'm very appreciative of everyone who's been a part of that community. Outside of the lab, there's been a lot of people that have encouraged me and really kept me going. And so I really have to thank, all of my soccer friends, all of my other friends who've come to visit us and gone on adventures with us. And it's really those things outside of lab that makes the hard days in lab so much easier. So I'm very appreciative of all of the friends that I have. This year has definitely been a bit of a hard year, but I'm so appreciative of my family, and they of course love to come visit us when Disneyland was open, but I'm just so appreciative of them for always being there, and even though they maybe don't always know or understand what I'm doing, I'm very appreciative for their continued love and support in really all aspects of my life. And the other person I really need to thank is my PI, Marion. I didn't imagine starting grad school that I would end up with a boss that I absolutely loved and adored and looked up to more than really anyone else in the world. But it's been an absolute privilege to really go on this adventure with her, and I've learned so much from her, both at the bench, but also just in life. And I'm so appreciative to have her as my PI. And the last person I need to thank is sitting here next to me right now, but that's my husband, Tyler. And he's really been, my adventure buddy, but my rock and everything that we have going on. And just all of the ups and downs, I really couldn't imagine doing anything without him. So with that, I want to thank you guys for your attention, and remind you that we do have a Zoom reception of sorts tonight at 5 p.m. And I'd be happy to take any of your questions. Thank you, guys.
Amber, that was great. Actually, kind of right on time. That's wonderful. I am going to, you have 2 questions in the chat. I'm going to ask them while other people, after that, if there are other questions, people can just unmute themselves. The first question you receive is from Chris Hughes. He said, beautiful work. Question for Amber. When you treat colon cancer organoids with low glutamine, do you block Warburg metabolism? And do you then see changes in oxphos versus glycolysis?
Yeah, that's a very good question. That's a little bit outside the scope of what we really investigated, because we're looking at kind of independent of the actual metabolic effect, is this kind of outside effect of alpha ketoglutarate. So I imagine there's has to be effects of Warburg metabolism and more metabolism focused things, but that was kind of outside the scope of what we investigated. But there's got to be effects in my mind.
The second question is from one of your committee members. Can you please explain your irradiation model and how it relates to human life?
Yeah, so I guess in some sense, it does feel like this irradiation model is perhaps a bit artificial, but there is definitely examples in human life where this type of wounding does occur. One, there are severe cases where people do get exposed to high amounts of irradiation. You can think of nuclear accidents or warfare, where you can see these effects happen to the crypt. The more common example is actually with cancer therapy, people that are undergoing local radiation for maybe different pelvic cancers, and as an effect, they'll actually impact the crypt. That's usually not as severe of an irradiation, but it can cause this effect where you have damage to the stem cells, damage to the proliferating cells, and then the crypt is able to return back to homeostasis. So it does relate to real life, but it is a little bit of an artificial system we use to really study the dynamics of the crypt and recovery process.
Okay. And I should say, while I'm asking these, if any of you want, probe a little bit further or back and forth, just, interrupt me. Another one of your committee members, Yang Shengqi. What is the evidence that RNA processing changes are due to protein modifications?
Yeah, so this is really one of the things I want to investigate further and really see if that could be one of the driving effects in the crypt system. And there are some examples of where you can have a phosphorylation change on an RNA processing regulator that can then cause downstream effects in RNA processing. So there definitely are some documented examples. And I think looking at some of those examples and how rapid the time scale is of those, it's very consistent with the rapid time scale we're looking at in the crypt setting. So I do think there is definitely some evidence that changes in protein modification could be really the driving force for changes in RNA processing.
Gina Lee asks, have you tried glutamine or glutaminase inhibitors in comparison with alpha KG treatment?
That's a good question. We haven't tried that. And that's definitely a good question to see if you're having kind of the exact same effects go on, or if there's something different with just altering the actual level of alpha ketoglutarate. So we haven't tried that, but that would be interesting to pursue and investigate.
Her follow-up is, what is the normal colon response to low versus high alpha ketoglutarate treatment?
Yeah, so that's actually a good question. So 2 answers to that. One, we did do experiments where we treated just wild type mice with high amounts of alpha ketoglutarate, and we don't see any biological impact. So there doesn't seem to be any damaging response. And in some sense, those mice actually looked a bit healthier than untreated mice. But we also looked on the organoids, and we started to do some of these experiments really in anticipation that that would be something reviewers would want. And surprisingly, reviewers didn't want that. And so we ended up stopping those experiments and focusing on some other things. So we never really finished looking at just an organoid-centric response to that high alpha ketoglutarate treatment. So that could definitely be something we pursue and look at a bit further.
Anand Ganesan asks, how prevalent is the shift in RNA processing in general during differentiation in other tissue types? Did you see any evidence of other mechanisms of immediate responses that might suppress expression of RNAs, antisense RNAs, for example?
I almost wonder if Marion paid Anon for this question, because this is something that we've really discussed in depth, is this idea that, maybe this isn't something specific to the intestine, although we have, this fantastic loss of stemness and differentiation process that happens there. But maybe this happens in skin. Maybe Anon, you can look in the skin setting. But that's the idea, is that this might be a general phenomena and a way to rapidly facilitate loss of stemness via RNA processing. So we don't necessarily yet have evidence that it's a more global effect, other than maybe looking at examples of previously published research looking at embryonic stem cells. But that's kind of our hypothesis, is that this is a more broad phenomena. In terms of the other question about looking at some of the other antisense RNAs, for example, we did look more broadly at other types of RNA processing. So I was kind of focused on skipped exon and alternate polydenylation. We did look at other types of RNA processing, but never looked, in depth at perhaps antisense RNAs.
MicroRNAs or anything.
Yeah. So that could definitely be something we could follow up and look at further to see if there is an overlapping effect there as well.
Yangsheng says, is it known that differentiated cells can revert back to stem cells in the crypt?
Yeah, so yes, it is generally known that there is that ability to de-differentiate. A lot of the experiments that have been done have been lineage tracing models. So they might look at a certain enterocyte marker and stain or mark those cells, irradiate the crypts, and then see if that marker will eventually label an entire crypt. And the idea behind that is, if you can have a label an enterocyte, it would then de-differentiate, become a stem cell, then give rise to an entire crypt and label an entire crypt. So that's been done for enterocytes, goblet cells, a variety of different cells, and really been suggested that all cells in the crypt have that ability. But the debate has kind of been what causes which cell to do that. And no one's really looked in an unbiased manner to see what's most likely. But it is, I think at this point, pretty well accepted that differentiated cells are that plastic.
Okay, Selma, great talk. Regarding the ABS lineage, the absorptive lineage that possibly reverts back to a stem population, what are the signaling pathways potentially controlling this? Do you have a clue from the transcriptome?
Yes. So the one thing, is you can probably imagine, we definitely wanted to look at right away was Wnt signaling. And you'll notice Wnt signaling was a little bit absent from my talk, which is abnormal for a Waterman lab person. But we were surprised that even within that 4-day window, we didn't see an increase in Wnt signaling. We did really look to see, is there just any changes in general ability to have a Wnt signal? And it does look like some of the absorptive cells have a little bit more of a potential to have a Wnt signal, but there's just not any strong Wnt signaling. And so kind of what we're thinking is that you need to reside at that base of the crypt for a longer period of time in order to actually get some of those appropriate signals. And one of the things that we just don't have the ability to look at, in our data set, and hopefully someone else follows up with, is how damaged is the stroma around that base of the crypt that's really the feeder of those Wnt ligands. Does it take the cell to reside for a while? The stroma has to repair and then start secreting Wnt to then facilitate that recovery and take time. So that's kind of outside the scope of what we know, but I still think Wnt is really going to be one of those important cues to regaining stemness. And aside from that, there wasn't really any other huge emerging signaling pathways that we saw within that 4-day window.
Okay, you should probably look in the chat, Amber. You're getting a lot of well wishes and kudos.
Oh, thank you.
Is there any other questions before we adjourn from the audience? Oh, Derek Reynolds, great work. It has really taken some twists and turns since I last saw it. How did you determine the amount of wounding that still leads to recovery?
Yeah. So we chose 12 gray X-ray irradiation for this experiment, and that was really based off of what people had previously published. And that 12 gray is kind of the maximum amount of irradiation you can expose a whole body of a mouse to and have them survive reliably for 4 days. Once you go past that, you start getting too much damage, and the mouse is overcome with that and can't recover. So that's been something that a lot of people have kind of looked at to try to figure out what the right amount is, but this is really kind of the max sublethal dose that's reliable.
I'll just read out one. Please send Stanley Behrens' congratulations, Amber, and best wishes to you. He's very excited and wish you much success.
Thank you, Stanley.
I don't know who's asking this, but is there a kombucha with high glutamate?
I don't know on that. I will say high glutamine and high alpha ketoglutarate are very tied with like high levels of protein in food. So things that have a lot of dairy and protein, or even like leafy greens, have a lot of glutamine. So, and it's possible there's something with high kombucha or in kombucha. So definitely look into that. Thanks, Mom and Dad.
Okay, Dickie, I'm not sure. You said this is a question from Arts. Do you want to unmute? And I'm not sure I understand.
No, I was just telling you that the question that you didn't know.
The kombucha.
Okay. Anna Chu from Arts.
Oh, yes, I didn't recognize the name of the sender. I apologize for that. Yes.
But actually, and I'll follow up on one more thing with that. That's actually kind of interesting. Is for a while, people have thought, like if they have cancer, that they might try to have a certain diet in order to aid in fighting that cancer. And people have thought, okay, let's have a low glutamine diet, thinking that cells are glutamine addicted. If you have a low glutamine diet, you might be starving the cancer. So this idea that maybe that's not the best idea, maybe you want the inverse of a higher glutamine diet, is definitely something to think about. And I know May Kong's lab is really following up on this further of, what's the best thing to feed or not feed a cancer cell. So there's definitely more work to be done on that. So stay tuned to the Kong lab for that.
Yeah, May just presented a sort of really bold alternative way to think about this at one of our cancer center retreats. So she's definitely focused on this as trying to convert this discovery into something that could be, a therapy perhaps on the road. Are there any other questions? How about MSG used for cooking? Monosodium glutamate, that can be a major glutamate source.
Oh, I don't know if I have a strong answer to that. That's not something I've really looked into, so I don't want to speculate too much. But in theory, if it's something that can convert into glutamine and feed into that pathway, it's possible.
Interesting. I think the question that Gina asked, which is what happens with high alpha Kg or high glutamate in a normal intestine, even though you overtly, morphology-wise, you didn't see any difference, but I'm sure, it also will be changing the microbiome if you have something high.
Yeah.
But that's a really interesting question.
Yeah.
Okay, I think I should call it quits. The committee, we use a different link for a breakout room where they're going to further grill Amber or not, I don't know. And if the outcome is positive, we will be celebrating at 5 p.m. on Zoom using this same exact link. So if you can make it, we'd love for you to join us. And thank you again, Amber.
Yeah, thank you guys.